Okay, hello everyone. Welcome to the first lecture of the special lecture series. So my name is Min Zhang, and I'm a professor of statistics and associate director of data science at Purdue University Center for Cancer Research. So this seminar series is part of our NCI-funded R25 summer workshop on big data training for cancer research. So today we have three panelists, John Fry, Nadia Lama, and myself. So John has helped with setting up the Zoom meeting and handling all the registrations. So he also records the lecture and posts the recording at our workshop website later. Now I would like to introduce our webinar moderator, Dr. Nadia Lama. So Dr. Lama is a research assistant professor in the Department of Comparative Pathobiology at Purdue. She has been the manager of the Purdue site of Collaborative Core for Cancer Bioinformatics since 2015. So Dr. Lama has a background in biochemistry, molecular biology, and computational biology, and she got her PhD in 2015 from Purdue. And her research interests include single-cell data analysis and data integration with a focus on immunological diseases of the prostate. So with that, I will hand it over to Nadia.
Thanks, Min. Hello and welcome. So just a couple housekeeping notes. So we're going to keep your audio muted for the duration of the seminar. Please feel free to ask questions at any time using the Q&A function, which is located at the bottom of your screen. And you can also upvote questions that you're interested in having answered, and those questions will float to the top and have a better chance of being answered because we'll ask those first. Now I'll hand it back to Min.
Thanks, Nadia. So it's my great pleasure to introduce Dr. Rebecca Doerge, the gleaned Dean of Mellon College of Science at Carnegie Mellon University. So prior to joining the Department of Statistics and Data Science and the Department of Biology at Carnegie Mellon, Dr. Doerge was the Trent and Judith Anderson Distinguished Professor of Statistics at Purdue University. Dr. Doerge joined Purdue in 1995 and held a joint appointment between the College of Agriculture and the College of Science until she left Purdue. So her research program really focusing on statistical bioinformatics to try to answer and disseminate biologically interesting information to understand the ultimate function of DNA and epigenomic associations. So Dr. Doerge is a recipient of many awards when she was at Purdue, including the Teaching for Tomorrow Award of Purdue University, the University Scholar Award, and the Provost Award for Outstanding Graduate Faculty Mentor at Purdue in 2010. So she's an elected fellow of the American Statistical Association, an elected fellow of the American Association for the Advancement of Science, and a fellow of the Committee on Institutional Cooperation. And she's also the chair of the American Association for the Advancement of Science, Section U in Statistics. She has published over 140 papers and graduated over 26 PhD students. So Dr. Doerge is past member of the Board of Trustees of both the National Institute of Statistical Sciences and the Mathematical Bioscience Institute. And currently, she's a member of the Engineering External Review Committee, the Lawrence Livermore National Lab, and also on the Danforth Plant Science Center Scientific Advisory Board in St. Louis. So today, we are going to hear an exciting story about the future of AI-driven science from Dr. Doerge. So please join me in welcoming Dr. Doerge.
There we go. I'm unmuted. It's a new drinking game. Every time you need to unmute yourself while you're talking, then you just take a drink. Hi, everybody. I'm Rebecca. I understand that this is very conversational, so that's exactly what we're going to do. I have some exciting work to tell you about that's ongoing at Carnegie Mellon University. Because I have what seems like an unlimited amount of time. I have an hour and a half. I probably won't use it all, but you never know. If you have short questions during my talk, please put them in the question and answers, and Min and or Nadia will interrupt me and ask your short question. If you have long philosophical questions, just hold until the end and we'll have a discussion then, okay? You can still put them in the question and answers, but we'll hold them till the end. So I'm Rebecca, and at Carnegie Mellon, we have been worried about the future of science, and that's pretty much the goal of what I want to talk to you about today. Those of you who know me know, so I was at Purdue University in the Department of Statistics and the Department of Agronomy for 21 years. And those of you who know me have seen this picture before. I spent the large portion of my career chasing the central dogma. Genes are transcribed and then translated into proteins. Over the course of my career and Min and Nadia's career, the technologies changed to sequence genomes, to transcript, to look at the transcript, and also protein quantification. So I'm a statistician by training, and made a career of chasing technology to figure out how to design experiments and analyze the data that come from new technologies. And so there are genomic hypotheses that are attached to, and certainly epigenomic hypotheses that are attached to the different levels of the central dogma and new technologies. And going back, the very first in my career 30 years ago, quantitative trait locus analysis took a genotype of a genetic marker and the statistical methodologies associated it to the variation of the phenotype. And here we're showing a phenotype of a fly or a Drosophila. And then microarrays came around, and then believe it or not, there were protein microarrays. They were super hard because proteins are three-dimensional. And then next generation sequencing came. So each stage of my career, we chased these technologies. And then right when I left Purdue, we were doing a lot of single cell RNA and DNA work. And so, you know, over the course of my career, the technologies changed and I've adapted with it. The phenotype here, as soon as I got to Carnegie Mellon in 2016, I was contacted by the Robotics Institute, and they were phenotyping sorghum plants in Ethiopia with a robot. And they wanted my assistance in understanding basically the statistical analysis behind QTL analysis. But I think I was on campus at Carnegie Mellon for about 2 months before they came to me and said, hey, we're going to use robots. So that sort of gives you the tone of Carnegie Mellon University. And over the course of my career, there have been common themes. The technologies change, things get faster, cheaper, and along with that, the data are bigger and the data sets become more complex. The basic biological questions are the same, but as the technology changes and the instrument change, the questions are the same, but they're at a deeper level. And so we can explore the data, or we can ask questions of the data using inferential statistics. The statistical challenges really haven't changed over the years. People still complain about small sample size, lack of replication. If they did replicate, then the scientists may have replicated in the wrong place. There's sampling variation, there's multiple testing, like asking the same question over and over again across your genome. And then once you manage to do all of this, you get a data set, you analyze it, someone else tries to reproduce your results, and they're not reproducible. So there's lots of frustrations that we've seen in these sorts of data across my career, Min's career, Nadia's career. Bad data, no replications, lots of pretty pictures, but the stories change according to the pretty picture. And the results, as I mentioned before, are not reproducible. And then the data are not shared, or they're not accurate, or they're not available. So these are all common themes that still exist in a large fashion. So if we fast forward, this is what I just told you about: the 90s, genetic markers, gene expression in the 2000s, sequencing technologies in the 2010s. And then as we are, you know, headed into 2020, which we all sort of want to forget that year, automation, artificial intelligence, robotics, machine learning, active learning are all informing how we're doing our science. And what I want to talk about today is remote controlled laboratories. So new tools and instruments are collecting massive amounts of data in every aspect of our lives, right? So we all, lots of people wear Apple Watches. We're collecting data at stoplights. Every aspect of our lives, massive amounts of data are being collected. And powerful artificial intelligence technologies are analyzing those collected data and making decisions about certain things like stoplights, when they turn red and when they turn green and yellow, and various other things in our lives. To do this, people are working and training across disciplinary boundaries, and things are definitely changing how we actually operate as scientists. So the thing here is that much of science research is currently limited by the speed and the scale of data that can be acquired. So if you think about how science is done, not what science is being done or what questions are being asked, but how science is being done, very little's changed in the last 100+ years. We are still doing science in the same way. We have an instrument, we have it in the laboratory, or we can go to a facility, we collect data, and then we analyze it. But a lot of this is still very human intensive, very labor intensive. And I know that we have some graduate students and postdocs that certainly noticed how many hours they've spent at the bench pipetting and doing repetitive tasks. So this is what we want to change in science. We want to change how science is done, the process of science. So machines are emerging that are transforming our work and our play and the fundamental fabric of economy. Self-driving cars, cell phones. I mean, has anybody ever thought about why hasn't science, how we've done science, why hasn't it evolved at the same rate as a cell phone, right? We're all happy standing at the bench pipetting. So why hasn't that happened? And like all things technology, there's a promise of greatness, but there's also a promise of social disruption. So machine learning, artificial intelligence, autonomous laboratories, that's the focus of today. If you are unaware of Carnegie Mellon University, it's really an interesting, very small university. We have 7,000 undergraduates, 7,000 graduate students as of 2 years ago. That's the first time they were equal. It's pretty much known as an undergraduate education that does big research. Andrew Carnegie in the 1900s founded the Carnegie Technical Schools for the purpose of educating the sons and daughters of the steelworkers in Pittsburgh. And that was the Carnegie Institute, they called it the Carnegie Institute of Technology, CIT. In 1967, CIT merged with the Mellon Institute of Industrial Sciences, creating Carnegie Mellon University. So Carnegie Mellon University has only been around since 1968, which is amazing. And so going forward, understanding that, the first Robotics Institute was founded at Carnegie Mellon in 1979. The Software Engineering Institute was founded in '84. And the first college of computer science was founded in 1988. And today, CMU is the youngest of the top 25 universities in the United States. It only came into existence in 1968. So the story I'm about to tell you is about automated science and Carnegie Mellon University's big bet that they're placing on automation of the process of science. So we are proposing the first academic automated remote controlled laboratory. This is a partnership with Emerald Cloud Lab, which is in South San Francisco. It was founded by 2 alumni of the College of Science at Carnegie Mellon. And they approached me and said they wanted Carnegie Mellon to host the first academic remote controlled laboratory for biology and chemistry. So this is a revolutionizing concept. It revolutionizes access to instrumentation, research, teaching, the definition of open science, and how we collaborate. Min and Nadia, are we okay on questions? Okay. So why would Carnegie Mellon do this? Well, we've been first movers on a lot of things, robotics, computer science, and they feel that this is the next big thing, that it will invent new disciplines, Emerald Cloud Lab is the industry dealer in this, leader in this space. And we really want to set the course of action for science, robotics, AI, machine learning worldwide, specifically by leveraging the strengths of Carnegie Mellon and the culture of Carnegie Mellon. So this is an artist's rendering of a cloud lab. You will see that there are very few people, and the people that are in here are up on the walkways. This is a remote controlled laboratory. It consists of automated instruments and robots. It is driven by artificial intelligence technologies. Every decision and everything about the environment is recorded and traceable, and it works 24/7. The instruments, I always tease that, you know, students and faculty and postdocs need to be fed, watered, and rest. So this instrument is 24, this instrument lab is 24/7. So what is a cloud lab? It's a remotely operated research facility that handles, without the user ever stepping foot in the laboratory, all aspects of the daily lab work: method design, materials, logistics, sample preparation, and so on. So you can design an experiment in your office at wherever you are, or on the beach, or wherever you're sitting. You can design your experiment, you can run your experiment, you can organize and collect your data, and explore and analyze and share the results all remotely. You never have to step foot in the actual laboratory. This relies on robotic instrumentation, and it automates routine tasks, routine data collection. The data are very large because they run on 24/7 schedules. But the nice thing here is that it combines machine learning and artificial intelligence. So I will show you an example that goes with this picture at the top here in a few slides. Using machine learning and active learning, as you do your experiments, the things are being learned from the actual experiment that can actually redirect the experiments themselves. So I'll show you in a little bit what I mean with that. These are self-driving instruments, and it's remote access, as I mentioned before. So what I want to show you now, and these are Rebecca notes to tell myself to stop sharing one screen and to share another screen. So I am going to, of course, I messed it up immediately. I'm going to stop sharing this screen, and I'm going to go here. And I have a video of and Nadia, I need you to confirm that you can actually see this.
As chemists and biologists,
Good.
we've always been firmly
yet,
bound to
no.
the laboratory. For us, the scope and limitations of scientific exploration have been defined by the instruments we have, and the scale of our work.
Not yet, huh?
No, you might need to
Sure.
maybe try turning
Hold
on
on,
screen
folks.
sharing again.
Yep, hold on. So I'm going to do that. Hold on, I need to. See, this is why I left myself notes. All right, can you see that?
Yes.
All right, and here we go, folks.
As chemists and biologists, we've always been firmly bound to the laboratory. For us, the scope and limitations of scientific exploration have been defined by the instruments we have, and the scale of our work has been metered by the long hours required at the bench. It's time to change that. Emerald Cloud Lab is a remote-controlled life science laboratory that allows scientists to execute their experiments without being anchored to a physical lab. In a cloud lab, experiments are driven by issuing commands over the internet, which are then run in a vast, highly automated central facility. With an ECL account, you have full control over every aspect of how your experiments are conducted. Control the transfers of volumes from less than a microliter to 20 liters. and the transfers of solids with masses from micrograms to kilograms. There are over 150 models of best-in-class instrumentation online at the ECL. ECL facilities run your experiments on demand 24 hours a day, 7 days a week, 365 days a year, leaving just hours between the moment you conceive your experiment and the moment you receive your results. It's not unusual for an ECL user to be orchestrating dozens of protocols simultaneously, far more than one could ever manage in a traditional laboratory. When you're ready, you can build scripts which automatically execute a series of experiments of arbitrary complexity, reproduce results, will process the data and generate reports for you to analyze. As chemists and biologists, our minds are capable of moving faster and further than the laboratory has ever allowed us. Take your seat in the command center. Transcend the lab.
And I'm back, right? Yay, good job. Okay, so that, I've talked about automated cloud labs for over 2-3 years now. And the 2-minute video saves a lot of time because the number of scientists that have said to me, oh, we have automation in our lab. So we're not talking about one instrument that's connected to a desktop that's connected to a database. We're talking about everything in the laboratory. I see questions and answers. Are those real or?
Yeah, they're legitimate questions. I don't know. Some of them are a little bit, you might want to save them till the end. They're not super quick, but if you want, we can get to them now.
Oh, all I see is money. Yeah, we're going to talk about money at the end. So ask that question at the end. Yeah, ask that question at the end about money, salaries in the future. And you mentioned that science is undergoing a very sharp shift. My experience, it seems like a bottleneck. Yep, we'll ask all these at the end. Great. Okay, so what does the cloud, it delivers a lot of things. Okay, so the wet lab folks, you're thinking, oh my God, 24/7, 365 days a year, that's fantastic, right? But what I really like about this is that it democratizes access, it democratizes science. And I'm going to say this more than once. I'm sure that I know that I have colleagues all over the world that are much, much brighter than me, much faster, much smarter, but they don't have access to instrumentation. They don't have the resources. When you come up with an idea for an experiment, and then you actually want to implement it or write a grant, you typically have to work with the instruments that you have available to you or the instruments that you can afford to buy on the grant. Never has an academic been able to have access to this. This slide says 180 pieces of instrumentation. It's well over 200 now. But imagine where you're not confined by the instruments. You're only confined by human ingenuity. And it doesn't matter which university you're at or which high school you're at. You have access to every instrument. It's mind-blowing. So it's the democratization of science that really it's my favorite part about this whole thing. It's high throughput, 24/7, we mentioned that, but it also parallelizes science, right? Science is a very linear thing, unless you're lucky enough to have 6 postdocs working in order, right? Which never happens. So this parallelizes science. So not only is it more efficient, more accurate, but it's faster in the sense that you can do simultaneous things and then bring them together. And then, as I mentioned before, this brings experimental science into the information age. Everything is automated, everything is recorded, everything is accessible. So this really does revolutionize and modernize science. Let's see here, where am I? My slides aren't changing. Woo. Okay, so when we think about, so the white circles here, data analysis, interpretation, and presentation, scientific ideation, and experimental design. That's really the human part, right? And this is when you think about automated science, you're dealing with inventory and waste management. And I've learned so much more about, they have these giant elevators that store samples and materials and things. Equipment maintenance and repair. Every instrument in the facility is tracked according to age, cycle, and if something falls outside the bounds of the machine, it's immediately flagged and the experiment can be stopped, or you're aware of what the new variation is. I'm a statistician, so I think about variation. The manual labor is totally minimized, and I can talk to you later about exactly how many people it takes to staff one of these facilities. And all data are recorded and cataloged and kept. So really, these darker circles over here, you know, it's much more than just doing your science faster. There's efficiency at every single step of this. So we're bringing together computation, data analysis, foundational sciences at an accelerated pace. Scientists are able to study very complex systems in a much more organized and refined way. But the bottom line here is that it's all dependent upon algorithms that are written by humans, artificial intelligence, where decisions are made that we might not know or they might not get recorded, machine learning, deep learning. You know, there's a lot of pieces going into this that we have to be very aware of. So, you know, COVID actually helped us a lot. You know, I did a lot of talking to the upper administration at Carnegie Mellon. This is the next bet. This is the next greatest thing. And then COVID hit, and I kept saying, you know, we probably wouldn't have had to shut down half of the science at Carnegie Mellon. This is the whole university, if we had had a cloud lab. And so, you know, pressing human challenges, global energy, climate science, it goes on and on. As I talk more about this cloud lab, imagine a series of cloud labs around the world that all talk to each other. So COVID's the perfect example. If we had a series of these cloud labs around the world, we could have brought the biggest and best minds to task on COVID, and they could have talked to each other through these laboratories. So this is an example of drug discovery, a very simple one. It's proof of concept. It's actually published. It's by Bob Murphy in computational biology at Carnegie Mellon. And I actually have notes here because I always get the numbers wrong. So let me get the, okay. So this is a drug experiment. Here we go. Okay. So we have a random access. This is a microscope that measures the protein changes given the protein change its location with a given drug. Okay, and the microscope is used to look to see what the protein's doing. So there's 48 tagged proteins. And so this is drug discovery, 48 tagged proteins and 48 drugs. So this is a 96 by 96 matrix. So that's 9,216 possible drug protein combinations, okay? And what this experiment is doing, so you have this random access robot experimenter, it generates raw data. The data are analyzed, and then a model is built. Things are learned from that model. New experiments are performed, and so on. Okay, so we know that we have 9,216 possible combinations of drugs and proteins, right? That takes a lot of time, and it's pretty expensive. Using active learning, AI, after doing 28% of the possible experiments, the model was 92% accurate. and 40% more accurate than would have been done if you just like randomly chose the experiments and went through all of them. So this graphic, the fraction of experiments performed and the accuracy of the model, this is just random choice, or you could just, you know, you're going to do all of them eventually, right? And you can see that active learning, you're learning, basically, if you do a protein and a drug combination and it's not going to work, then anything with those sorts of features isn't going to work. So don't bother doing them. So you can see in this graphic over here, the white was the number of experiments, the correct predictions are green, and the incorrect predictions are purple. But you can see here, right, a large portion of these are purple, so they didn't even bother doing those. So this is so cool because it's faster, it's efficient, and you're actually learning as you go along. So when we go back to, this is just one this is an old example, but it really wraps your head around it, because if you then go back to an automated lab and you have the ability to learn from your data as you go, you can direct your experiments in a certain, you know, you're not going to waste a lot of time doing a lot of things that aren't going to work. That's the best way. And as a statistician, I have had a hard time in the beginning. I'm like, yeah, but you need, you know, you can't direct your experiments, but basically you're taking away the things that aren't going to work and you're focusing on the pieces that are going to give you information. So my aside on artificial intelligence, I need to have an aside on it because I think Nadia's heard me get off on my tangent about this. Artificial intelligence has no theoretical basis. Okay, this gets lots of computer scientists very upset, but in the end, they say, you're kind of right, Rebecca. So because it has no theoretical basis, mistakes can be, you know, everything's algorithm based, right? And then you turn this thing loose to explore, right? And mistakes can be made in the algorithms because we're only human. And then artificial intelligence goes from there. So humans, you know, programmers, software engineers, computer scientists, they write the first set of instructions, which inherently is based upon their knowledge or what we know in the world so far. And we've all seen in the newspaper the biases, right? The human face recognition that were built on, you know, white Northern European faces, and they neglect to recognize African faces or Asian faces, right? We've all seen that in the newspaper. That's exactly what we're talking about here. So while I am all on board for automated science, I am very aware of the power of artificial intelligence and the need for really understanding the theoretical basis for it. Okay. So I asked some colleagues to give me some examples about AI and why they were worried about it, the black box of AI. And the first colleague said, if I consent to my insurance company having my genetic information or someday my protein or metabolic information, can they use data mining, artificial intelligence, to change my cousin's premiums without consent? So they learn from me and then it impacts someone else. So that's the black box model of AI. A second colleague at Carnegie Mellon who works in brain-computer interface, so these are neural probes that for people who are not able to move their hands or their legs or they're paralyzed, they put these probes in the brain and then they can think about moving their arm or doing something and it happens. So this person describes, you know, the manipulation of physical objects. And it says that this is currently geared towards helping people with physical disabilities, but it's easy to envision this growing beyond those applications. It feels like there's some legal ramifications once people are able to more directly manipulate environments with just thought. So, you know, as I mentioned a few slides back, there's good and bad that happens with all of these advances. So the take home here that my aside, there's a lack of transparency about artificial intelligence. The lack of transparency is what we don't know, but then also the decisions that are made that aren't tracked. We need to understand the limitations and we need to open the box, the black box lid. And so this just brings home that all design actions, decisions using the cloud lab are recorded and reproducible. So we're going to talk about open science eventually in a little bit. So these are shared, the transformation is that these are shared laboratory facilities. In a place like a university, this gets rid of every lab having to own their own machine. There will always be days where somebody needs to own a machine because they need to tweak it, but this is a shared resource laboratory facility. It increases the pace of work. It's training the next generation of scientists, which I know we have a question about. And the experimentation is not limited to access. And this goes back to the democratization of science. We're able to address big societal issues and shift rapidly. So again, COVID, if we had access to these labs and we were just doing, academic science, we could have pivoted literally overnight and started working on COVID. So I'm kind of dragging you through all the talking that I've done at Carnegie Mellon. And one of the questions basically was, Rebecca, if we build it, we need to know that they're going to come before we build it. So, why would they want to use this? So as I mentioned, it increases the capabilities. You have access to every instrument available to biology and chemistry. I also want to say, do I think that all of science is automatable? No. I think that half of the science going on at Carnegie Mellon is automatable across the whole campus. There, of course, is science that you cannot automate. I'm not saying that we should automate the whole thing. There's increased productivity. So based upon the Emerald Cloud Lab customers, which are mostly pharmaceutical companies, the pharmaceutical companies do not need an entire laboratory. They don't need every instrument known to man the way academics might, but their research productivity increased 7 times just by having access to a cloud lab. The conservative estimate based upon NIH scoring rubric was that, I don't know that this is a good thing, but 2 times more research papers can be produced when using the automated lab. There's a cost of $15,000 per savings per research paper. Personally, I think that's a very low cost that might not include human capital. And then with respect to submitting grants to agencies, there might be a 20% increased proposal success because you're not, things are in an automated lab. So these are all very general estimates. Carnegie Mellon submitted their first NIH grant, which budgeted the cloud lab. Emerald Cloud Lab is allowing us to use their laboratory until our laboratory in Pittsburgh is built. So I'm very anxious to see the reviews of that particular grant, given that there's no money for instruments or anything having to do with an on-site Carnegie Mellon lab. And of course, training the next generation of scientists. So what's in store for the future of Emerald Cloud Lab? Right now, when I think a few slides back, I said there were 180 instruments. They're bringing that up to 250. And they are launching, this kind of blows my mind, they're launching a desktop version of what they call the command center. And they're developing this ecosystem across the world of cloud labs. That's good. So redefining instruction. So as I mentioned before, COVID really helped us because Carnegie Mellon is still remote. So I haven't been in my office in 16 months. And we asked for special exceptions for our introductory wet labs in biology and chemistry. They all had to be socially distanced. We had to do special cleaning. It was a real hassle. We have been training faculty, postdocs, and grad students on the Emerald Cloud Lab in San Francisco for over a year. And one of the faculty that we trained, Marcel Bruchet, he said, I'm going to teach my class using the Cloud Lab, an undergraduate class using the Cloud Lab. And I was like, here. So what I'm about to show you is results from an undergraduate course. So accessibility, adaptability, scalable, shareable, truly reproducible results. So anyone who's been in a wet lab knows that technique is a major barrier. We've all heard people who have good hands and bad hands, or gold hands or bad hands. This gets rid of all of that, right? You can have really bad bench hands and still be a great programmer, and you can do great science. So I really like that because I have bad hands. And then we know that the impact of variations from minor statistical issues, you know, like measuring things wrong or doing a variety of things that undergraduates do, can definitely change the results. And so this is just an example of student acquired data. And I looked at this, I'm like, is this one group of students? Is that another group of students? And I think it's two sections, maybe. This is one section and that's another section. And the data should look like this, okay? And I see a line down here, but I don't think that's a really bad instructor. I don't know what that line is. So the point of this is that the students, you know, good hand, bad hands, floppy, they don't necessarily have good experiences in the wet lab, right? They're not getting what they're supposed to for a variety of reasons. So the challenges of training bench scientists, and I'm kind of laughing at this because, you know, I was allowed in the lab when I was in grad student, and they pretty much said, don't bring her back. She's a danger to all of us. So I'm enjoying this. So the how and why matter. So if the results aren't previously known, technique must be consistent to draw a conclusion. There's a typo there. Students can't explore the content, the concepts and the strategies because they're too busy dealing with the noise. So low noise and reliable data sets are a much better experience. And then it just, automation breeds a better experience in that the data aren't as noisy, the data are collected properly, and it sort of builds positive habits for students. So, you know, there's no filtering, there's no outlier pruning, there's no p-value hunting, as we say, right? Because the data are just better and the whole experience is better. So this example in I think it's the organic chemistry class. So liquid handling robots with undergraduates. Never done it, but from what I understand from Marcel, this is not something that you want to do. That they, you know, it's an issue. So laboratory robots that bypass the mechanical barriers are a good thing. And the robotics are usually set up for specific needs, and they're expensive and difficult to implement. And you don't necessarily want a bunch of undergraduates in there at the same time. So this was, we piloted teaching a course on the Cloud Lab. It was COVID induced. And it not only demonstrates the possibility of using it for instruction, it encourages the students to go well beyond the experimental designed stuff that they're supposed to. So, and I like the fact that it takes wet lab and computation and puts them together and really teaches students these important skills. So the objective is to explore a lignin receptor binding by fluorescence activity. And so you can see here 100-fold activation of the fluorescence. Down here is the red line, okay? And so the students, this is an example workflow. The students make approximate test solutions online. They characterize them. They measure the concentration online. Using the characterization in subsequent experiments, they perform some other measurements. They analyze the data to estimate their parameters for the next experiment. Okay, they set up the next experiment with new parameters, and so it goes, right? This is all online. So if you're physically in a laboratory at any point here, a mistake can be made and the experiment blows apart, right? So this example workflow is really really brings it home. So each student builds their own protocol online. This is an example of a protocol. The Cloud Lab ensures perfect technique, which I really love that because there is no technique, you just have to code. The students can be compared across, they can make comparisons across distinct molecules, and multiple experiments and analyses can be sequenced in a single script. So this is really where the parallelization of science comes in, even in a teaching lab. So we were lucky enough to be able to use the Cloud Lab in San Francisco at Emerald Cloud. The lab equipment gathers and returns the data in a database, and the automated process provides data that are searchable, they're privacy covered, and programmable. It comes in a format that can go into a programmable environment. So this is actually a picture. This is I showed you artist renderings before, but this is actually a picture of the Cloud Lab in South San Francisco. So any observed differences are due to the dye used, not the operator. And the bottom line here, if you think back on the slide where the student results and the instructor results were vastly different, the student and the instructor results are indistinguishable. So this goes to show how technique is taken out of, the technique issue of doing wet lab science is taken out of it. And of course, everything's distributed and parallelizable. And then I'll skip this one, but Marcel, he drank the Kool-Aid, Marcel Bruchet. He pretty much has, I think he said 90% of his research translated to the automated lab. And what he's done here, so this is a layout of a automated facility, and it might be the one in San Francisco probably. And what he's done here is he's shown 3 different workflows, and he's shown how, so let's just go to the yellow workflow here. He's shown how it starts and then the path through the Cloud Lab that it takes to be finished. This is very, very cool, right? So here he details out exactly what's being done. So just, I looked at this slide and I'm like, imagine all the different paths that you could possibly make, right? If you had 250 instruments. So this design, this is the part of the lab in South San Francisco, and this is exactly what we want to do for Carnegie Mellon. So the promise of the Cloud Lab, distributed experimentation, resource optimization, shared data, immediate shared data. So the days of a postdoc leaving or a graduate student leaving and people can't remember where the data are or find the data, or you publish a paper, which now the data have to go with the paper, but the programs don't necessarily go with the paper. And the postdocs don't go with the paper, and the technique doesn't go with the paper, right? So this makes everything traceable, reproducible, right? And then the other part of this, so we have the research part, we have the teaching part, we have the democratization of science, putting in the hands of really smart people anything they need. The last thing I like about this is the open science. So in libraries, and Purdue is extremely well known for libraries, there's something going on called open science. So this is, sharing all results, all code, anything about it. So shareable data sets, sharing methods, and all kinds of things. So imagine, and I'm going to talk a little bit about open science, but imagine a day that you use an automated lab to do your experiment, you get your results, you write your paper, you submit your paper, your data, and the Cloud Lab parameter file to the journal. And so I can see a day using the automated Cloud Lab where no one questions your results because they have your automated parameter file. If they wanted to reproduce your results, there you go, just do everything that's already recorded in the parameter file. And I can see a day that people say, oh, the experiment was done on a Cloud Lab, let's just take the results and proceed, right? This gets rid of all the question marks about whether data are reproducible or not, which is really super exciting. So this goes to open science. And this is where I put my slides together at the last minute. So talented scientists everywhere aren't limited by the access to data, and shared protocols. So when I came to Carnegie Mellon, they said, look, you know, science isn't at the level of engineering and computer science. We need you to leverage the strengths of the university to raise the profile of science at Carnegie Mellon. Okay. And I think we're doing this because imagine that you have an engineer who makes an instrument, right? And then he just plugs that instrument into the cloud lab. Now everyone at Carnegie Mellon has access to that instrument, and all collaborators have access to the instrument. They call this a plug and play. They call it an app-based wet lab because you make an app and you plug it in, everybody has access to it. So we are very excited about collaborating with new partners. So we have campuses in Doha, Qatar, Rwanda. We have a campus in San Francisco. And so this allows us to work across our campuses. Our resources and certain science in Qatar are very hard to work with the Qatar Foundation. So using an automated cloud lab actually solves a lot of issues. The historically Black colleges and universities serving underrepresented populations. Again, this is about democratizing access, right? This no longer negatively affects where a student does their education. So on the open science side of things, we're embedding the best practices in open science from the beginning. So we have a repository, we have a secure platform. So we are working with libraries with respect to the results and the data that come off of this laboratory to really open data and open science up worldwide. All right, so closing thoughts. I know this is kind of crazy, and I just keep thinking I'm really glad that I had the video because it really does exist, and you know, we really are using it, but I know it's a game changer and I know it's a mind bender. Access to new instruments, plug and play, I mentioned that. Protocols are shared electronically. No longer do you have to go to someone's lab to learn a technique or learn a protocol. You can just get the code. Science is parallelizable, which brings efficiency and speed and all sorts of things. All the details of the experiment are captured. That goes to reproducibility. The decisions are driven by AI and machine learning. I shared my thoughts about AI with you. And I think that is something that we all need to work on, reproducibility, and then publishing results where one experiment ends, the next begins. So I'm just really excited. At Carnegie Mellon, I can tell you that and I will talk about money, question number one, I think, or two. We have the money to move forward. We are purchasing a building off campus. So the cool thing here is that this does not have to be in a shiny new building. It can sit anywhere on campus. I hear my front door opening. So we are in the process of buying a building. We are negotiating with instrument companies, and we will have the facility is designed, and we will have it up and running by early 2022. So stay tuned. Very excited about it. And I can tell you the details of how we did that. All right, so that's my end of my story. And I'm happy to take questions. So I'm going to stop sharing so that I can get back to full screen here. And I'm going to close my office door at home so that my dog doesn't bark. All right, Nadia, Min, any questions?
Yes. So we've got.
You are muted. No, you're not muted. Okay.
We've got a few that are a little bit longer, so I can read them.
Oh, I can read them.
Or if, yeah.
I'll just start at
the top.
I'll just start at the top.
Yeah.
Okay. So everybody can read with me. This is beyond amazing. It is mind-bending, I agree. But all I see is money. How do you see this becoming a reality in academic labs in the future? Of course, staff, students, and postdocs will still be needed and will be needed to be trained, so money cannot come from their salaries. Perhaps you will get to this. Yeah. So, and then Andrea says, and of course, this goes beyond initial instrument acquisition, but includes upkeep, service. Yeah. So, this is proof of concept, a big proof of concept. This is about a 50, if we have no donations from instrument companies, right? So if we think about this, we're training the next generation of scientists. Industry is going, they're going to be the first folks to use this. So we are training that group of students to go into industry. With respect to, I can easily see, and I know this is being recorded, but I can easily see NSF and NIH setting up these facilities to be utilized by academics. If you think about it, it would be so much cheaper for them to have, I don't know, 10 of these around the country than to buy everybody their own sequencer or buy everybody their own mass spec or whatever, right? So ultimately, I know it's a big number to think about alone, but when you think about it as a shared resource across the country or across the world, it actually is much more efficient. With respect to upkeep, maintenance, and things like that, for the Carnegie Mellon facility, it is a recharge facility, and I'll talk about money in a minute, but it's a recharge facility, which means it has to pay for itself. We worked with an MBA student who, funny enough, had a career in life sciences before he came back to get an MBA. And we will turn a profit after year 3. So it'll be $6 million loss in the first 3 years. And then we'll start to turn a profit in year 4. And after 10 years, we will recover the max number of $50 million. So if we get no cooperation from instrument companies and we have to buy everything outright, it will be $50 million. And that's what we have right now. But we fully expect instrument companies and pharmaceutical companies to want to work with us because we're training the next generation of scientists. So that, I think, covers the first question in detail. And the $50 million, that's just a round number. It could be 47, it could be 51. I just use 50. Next question. Ooh, let's see. You talk about democratizing science by this, but it's going to be effectively cornered by companies that have money. I don't really understand that question. Anybody want to interpret it for me? You talk about democratization of science by this technology, but it's going to be effectively cornered by companies that have money. I'm going to let that one sit for a little bit. You mentioned that science is undergoing a very slow shift away from the bench top science with an emphasis on directed hypothesis-driven research to an AI machine learning unbiased form of research. Oh, okay, I'm gonna hold that second. So we got feedback, Rushi. Academic science is struggling for funding, but this ultimately, if this is proof of concept at Carnegie Mellon, okay, but we're talking to the federal agencies and we're talking to Washington, I think that we can transform science worldwide, which will ultimately put money into the science and not so much into the hands of, so academic scientists are struggling for funding, but ultimately this will save money. Okay. All right. In my experience, this seems to be a bottleneck for progress that happens at the administrative where grant writing is based on preliminary data and favor hypothesis. Do you believe that this is an issue? If so, what do you think should be done to change it? I don't think I understand the question. Sorry. You mentioned that data is going away from slow shift from bench. Nadia, you want to More philosophical, curious question. Yeah. Now, do you want to interpret Emmett's question for me? Because I'm just not tracking.
So I think that
Or Min.
he's talking probably about in the beginning where you mentioned that, like science isn't keeping up with the I guess speed that technology is moving in terms of, you mentioned like the cell phone and stuff.
Yeah.
And
yeah.
I think he's saying that in his experience, it seems like part of the bottleneck for this is the administrative level, or maybe even funding agencies or study sections where they expect,
preliminary data.
Yeah.
And
hypothesis.
Yeah, Emmett, I think that, well, I agree with what you're saying. And when we talk about revolutionizing science, the whole thing's revolutionized. So I think that I think we'll always have hypothesis-driven science, but how we get to those questions will change because they might be generated by the science themselves, right? Which is going to get a lot of people upset. But yeah. Yep. Boy, this is jumping all over the place. More philosophical or curious question. How do you handle serendipity in such an environment? Yeah, this is a question, the random experiments, like, most drugs aren't actually used for what they're designed for. So I've talked to Marcel about this, Marcel Boucher, and he's the one, we have a lot of different scientists using this facility, but he says that you still get results, serendipitous results. Because it's not just the one-off. If it keeps happening, you're going to collect those data. So he maintains that this is not cookie cutter science, that you still get those random, consistent results that make you go, huh, what's going on? So you still get serendipity. As long as so Xing, no, who was the question was that? Sorry, just jumping all around.
Alicia.
So as long as you know what you did, so that's the thing, right? So there are many scientific experiments that people measure wrong or do something that they think they did that they actually didn't do, and that's the serendipity of it. So in that sense, no, that's not going to happen. There won't be human error that gives positive results that sometimes happen now. Yeah. Alex, this is exciting and I look forward to tracking the progress of remote labs. You can go to Emerald Cloud Lab website right now and it tells you all the instrumentation and it has that video on the front page that I showed you. I think it's an excellent idea and would love for the idea to take root and become commonplace. My question is, how would these resources be shared amongst hundreds of labs once this becomes popular? Even if a laboratory has their own dedicated cloud lab, it seems labs with more funding. So I'm not even going to finish this. So this is exactly like cluster computing. At Purdue University, there's cluster computing that not everybody needs their own server anymore. You come in, you buy nodes, and you have access to those nodes. So if I'm a new assistant professor, I buy 5 nodes for $5,000 each, whatever they are. And I have first access to those nodes. If I'm not using them, then other people have access to those nodes. So the cloud lab is organized in the comparison is it's called a thread. So instead of a node in computing, this is called a thread in the cloud lab. So I can see a day where an assistant professor comes to Carnegie Mellon and they buy X number of threads. And they have priority to those threads 24/7. But if they're not using them, then someone else has priority to their threads and the first person's threads. It's just like cluster computing. All right, really cool, at least for a subset of experiments. Remember, I said not all science is automatable. In vivo, any ideas? I have lots of really funny ideas that I don't want recorded, but yeah. Another question about the feel of the experiment. In my previous lab, we were trying to create an in vitro gel encapsulation of cells experiment. Lots of small things had to be standardized, had to be made sure that's close. Now we had 5, 6 people trying to do the experiment. Only one of us could successfully standardize it. Imagine doing this through robots. Yeah, I don't, I'm sure it can be, I don't know enough about the cloud lab to answer this question, but if you had 5 or 6 different threads all working at the same time to do the optimization or whatever needed to be done. I'm sure it could be done in the same way, but in vivo experiments, that's part of the half that's not automatable for right now, for sure. Can you show some real experiments done by an AI, if ever done? For example, PCR. I don't have any right here now. I can show you, but Chris Langmead from computational biology at Carnegie Mellon has published papers. Chris Langmead, Langmead. This is all, really new stuff. So, all right, how'd we do? Chris Langmead.
Good.
Oh yeah, Nadia put it in the.
Is that correct spelling?
Yep.
Perfect. That's what I thought, but. All right,
Yep.
yeah, you got through. Are there any other questions?
No open questions.
No.
And we have 36 people, which we started with 30. So usually when you end with 6 people and you started with 30, you know it didn't go very well. So I realize that there's lots of open questions on this. I hope that thanks, Alicia, I appreciate it. I hope that I gave you my task. My goal was to give you a 20,000 foot view of what's going on, to make you think about how science hasn't kept up, and make you think about how we can actually automate science universally to ultimately get more science done. So, and I know that there's AI issues and money issues and, people issues, but that's the goal. So, yeah, I mean, if you want me to come back and give an update next year, let me know, because supposedly in 2022, we're going to be up and running. So the last question, maybe I'll be able to show you real data from our lab. Oh, and I wanted to tell you one more thing. In my talk, I mentioned the number of people that it would take to run one of these facilities. So Carnegie Mellon, we put together a working group of scientists, and I said to them, make your wish, the working group was from all over campus. Make your wish list. This is what Emerald Cloud Lab can do right now. And this is about 1.5 years ago. Give me your wish list of instruments that we can start with. And I was thinking we could do 1/3 of the lab, and then 1/3 of the lab, and 1/3 of the lab. Turns out that, the materials handling and all of that, that's a big upfront expense. So we will start with about 75% of a total lab, and then add to it if needed, right? So there are certain parts that science doesn't go on at Carnegie Mellon, so we don't need it. But the other piece that I like about this a lot is that, like mass spec, right? If you have 4 mass specs and we think that that's going to be enough for Carnegie Mellon and it's not enough, we just plug in 1 mass spec. Instead of buying 5 mass specs for the whole university where everybody has their own, we just plug 1 in. And then, so this is what I really like about the utilization. We can add things to the utilization and take things out based upon usage. So, and anyway, back to the number of people it takes. We think that it will take 4 to 5 technicians to operate the Carnegie Mellon Cloud Lab. And these are not laboratory technicians like you guys are used to. This is, it really is a very interesting operation. Everything's barcoded, and pretty much a technician goes, punches in the number of the experiment, all these barcoded things come out, they take it over to this bench, they plug everything in and they press go. I wouldn't be surprised, I don't really want to insult anyone, but I think that they're well trained to run an automated lab, but I don't think that they have PhDs in biology and chemistry. So anyway, stay tuned.
We have one more question if you're.
You are muted.
Another question?
There we go. Oh, there's another question.
Yeah.
Follow up, Alex. Hi, Alex. You were here before. Follow up to the part about labs purchasing threads to gain priority in queue for experiments. Would this take away from the democratization aim of the cloud lab? And how would labs with less funding, underrepresented groups? So this is a good question, Alex. I fully expect for the CMU Cloud Lab to collaborate with undergraduate, underrepresented minority, majority populations, and we will dedicate a thread or however many threads to that specific university or PI or however. So that is, if you want to make something a priority, then you dedicate the threads to it, right? So, yeah. And oh, I know what I wanted to say, Alex. I also wanted to tell you that in Pittsburgh, the life sciences industry is very hot right now, very up and coming. We have a lot of startups in life sciences, and wet lab facilities are at a, premium. So we don't have a lot of wet lab facilities for startups to take advantage of. So we have saved, our business model saves 20% of the threads for life science in Pittsburgh. We want to support the region and we want to help science and life science. So 20% of the threads are saved out for entrepreneurial type work that is for profit. And I'm sure, we have the University of the UPMC, it's the University of Pittsburgh Medical Center, it's huge. I'm sure that we could sell every single thread that we have to UPMC. That's not the purpose. This is truly an academic endeavor. Somebody, I'm reading the chat. Loving this rant. I've had this, these are all out of context, but okay. All right.
That was on the AI.
Oh, good.
Rebecca on AI.
I bet your father's a computer scientist, Kristen. All right. Okay.
Okay, so if there are no, there's another one. Oh,
No.
Thanks for the great talk.
Thanks, everybody. Oh, yeah. Kristen says her dad is a computer scientist, unlike Nadia, whose father's a computer scientist. Who I know. Yeah, it's okay, Kristen. We're right. So I'm sure he argues with you, but he knows he's not totally right. Anyway, okay.
Okay, yeah. Thank you so much, Rebecca, for the wonderful talk. Yeah, I guess next year we'll still have this workshop about this time. So I think we are looking forward to see next.
No pressure, Rebecca. No pressure, Rebecca. Thank you, everybody, for your patience. I appreciate it.
Yeah. Okay. Thank you, everybody, for coming, and hope to see you tomorrow then. Bye.